right, guys, tonight's special guest speaker, Erick Ault, is a research administrator at the Florida Fish and Wildlife Conservation Commission's Tequesta Field Lab. Erick has more than 20 years of experience as a marine fisheries researcher, specializing in fish species that live out on our coral reefs and in our coastal waters. Erick and his team conduct a variety of fisheries-related research projects along the east coast of Florida. And tonight, Erick's going to give us a broad overview of the research work being conducted by FWC in our area, with a particular focus on some of our favorite gamefish species, but I think there's some other cool stars of tonight's presentation as well. It is with great pleasure that I introduce you guys to Erick Ault.
Thank you, brother. Can you guys hear me? Am I broadcasting? Awesome. Well, thanks so much for that great introduction, Zach. So what I was thinking about tonight was first talking to you a little bit about our lab, because it's kind of a neat story about how our lab came to be where it is, and then also go into some of the research that takes place at this lab, and then when we get to the stuff that I'm directly involved in, then I want to go into a little more detail, all right? Let's see here. No, my cue is back there. No. Well, I got the laser, but... All right. It's all right. We can work with this. Start right there. First off, how many of you folks are here year-round or in the room? Awesome. And then the rest of you guys are here. So I will tell you a little bit about myself while we're getting rolling, is that I am not native like a lot of you guys, like a lot of you folks are not native as well. But my family moved down here in the late 70s, and I've been in this area either in Palm Beach or in the Jupiter area since then, and I grew up with a passion for going fishing, being outside, doing things, and that's kind of how I got involved in this process, is that I wanted to do what I could to help preserve that and make it available to my kids, hopefully grandkids and stuff down the road. So that's kind of what kind of sparked my passion in going down this route. Well, you guys are familiar with Murphy's Law. Well, there's also that tech law, too. So it's a combination. Whatever can go wrong will go wrong. I'll tell you what's coming up, though, in my next slide, is whenever I tell people where I work, they usually always automatically assume that I am a member of our law enforcement team. And they probably, for the most part, it seems like most folks think that that's what we're all about. But what I wanted to do is kind of give you a little bit of the background of our agency, and then also kind of show you the different groups within our agency and how they interact. So first off, the Florida Fish and Wildlife Conservation Commission, or FWC, was established in 1999. It was a constitutional amendment that merged the Marine Fisheries Commission and the Game and Freshwater Fish Commission into one entity. It's made up of 7 commissioners that do most of the decision or management. They make most of those decisions in terms of how our policy is going to be managed. Those are governor appointees, and they typically serve a 5-year term, and they can serve multiple terms depending on the situation. We also have a whole series of administrative offices. There's a lot of them. I'm not going to tell you all of them, but things like the executive director, finance, legal, things of that. But once you drop down a layer from there, you get into the more specialized groups or divisions, and that's kind of where we fall in. And up first, we do have a law enforcement group. We also have a hunting and game. There's a freshwater fisheries management, a marine fisheries management, the Habitat and Species group, and then the group or the division that I'm in is the Fish and Wildlife Research Institute. Now this group here primarily is tasked with research and monitoring efforts aimed at gathering information to either put in science publications or reports, or aid decision makers in management strategies. And then further, the state's broken into 5 different regions, of which we're in the South region. And we have our main regional offices off of North Lake, just west of the Beeline Highway. And there's a lot of staff that work out of there, but also a lot of our management staff work out of there. So now, let's see. Boom. All right. So our lab, the lab that I operate, where it's at, is about as far as you can get south in Martin County. It's on the county line, the southeast corner. And it's a pretty interesting story. I had a bunch of our senior management come do a site visit, and I thought, hey, let me dig into how we got to have a lab here. And as I dug into the story, it was pretty freaking interesting. So I was like, man, let's check this out. So before I move on, what I want to show you is actually, this is southern Martin County where the lab is, and then this is a blow-up of the lab. But I want you to pay particular attention to this landmass, this Camp Murphy J.D. Park area, as well as this area here, Trapper Nelson and Jupiter Hills. All these kind of come in to play a part or play a role in the story about the lab. All right, so starting at the beginning, that chunk of land belonged to the U.S. military. And it was during World War II. And back then, the radar was a top secret technology, and that's where they were doing a lot of radar testing. So it was kind of neat, and I was just reading about it. There was a community out there. I don't know if any of you guys have ever been out to Jonathan Dickinson. It's a pretty nice park, pretty neat place to be. But there are still some buildings. But in its heyday, there were hundreds of buildings. There were, there was a movie theater. There were grocery stores. There was, all kind of things in there that really made it a nice community. But resources being what they were, once it was no longer, I guess necessary or as important as some of the other things going on during that time, they decommissioned that area and moved. A lot of those buildings were taken off-site and moved out, but some of them are still in place. So if you go out there, you can still see some of the buildings that were in place during that time. So they had this big chunk of land, and they didn't just want to turn it over and just let it sit. They thought, this is such a pretty area, we should turn it over to the state with the idea of the state making it into a state park. And in 1948, that's exactly what happened. They turned, that's when Jonathan Dickinson State Park opened. Now, it transferred to the state in 1948, opened in 1950. Some of the big amenities at the time, of course, were the Loxahatchee River, which has now been listed as a scenic river. But even back then, if you can imagine how cool it looks now, it probably looked 10 times cooler, back in those days. And then a homesteader by the name of Trapper Nelson, whose real name was Vincent Nostakovich, a guy from New Jersey, had set up a property, and he was known as kind of the wild man of the Loxahatchee. So he had this property. He had a wildlife zoo. He would wrestle alligators. He'd come swinging out and beating on his chest like Tarzan. It was pretty cool, and that was an attraction for folks that come to this park. Continuing on, so for a while I did some digging, and it was really cool to try to find. Now, remember, our lab is on the corner of US 1 and County Line Road, and this is that corner back in the 50s. And back in that day, US 1 was the main travel corridor. But Jupiter Tequesta area wasn't the destination that a lot of folks went to. It was a pass-through. Most folks were heading to Palm Beach, Fort Lauderdale, or Miami. Those were the hot tourist spots during that time. But, people would do what they could to try to capitalize on that tourist dollar, a lot like Florida nowadays. And I found this ad. So this building, there's still remnants, I think. There's still structures there. I'm not sure. It looks a little bit like this, but like some of the amenities back here, they had air conditioning was a big plus, color television, things like that, central heating. These were all things that would bring folks in. But for the longest time, this area just kind of sat undeveloped. Now, in the early 60s, Palm Beach County leased that corner, and in it they built a welcome center. And the idea was that they would promote all kind of touristy kind of attractions in Palm Beach County. And when I gave this presentation, one of the big things that this building was used, community stuff, Girl Scout meetings and that, is that they were known for serving people, giving you a glass of orange juice when you stopped in there. And a lot of people that grew up or spent time in that area back in that time remember this place for that. Now, when I mentioned the orange juice, it was then that some of my team members came out and presented everybody with glasses of orange juice. So at the time, it was a pretty cool event. But timing's everything. About that time is when the highway system was coming online, I-95 and the Turnpike. And with the event of that happening, it deterred or pulled a lot of the traffic that typically traveled that US 1 corridor out of there. So it kind of sat there, or it kind of was sat there for a while, and nobody was utilizing that property. Eventually, it wasn't maintained as a welcome center, and it wasn't being utilized as a community building either. So in the early 70s, a couple groups started utilizing that property. One was the Florida Marine Patrol, which at the time was under the Department of Natural Resources. And another was the Martin County Fire Department. Now, Martin County utilized this property as kind of a satellite fire station, and for the most part, they kept some of their show vehicles and some of their parade vehicles, but they built all their own fire trucks at the time, 6x. And I talked to some Martin County firefighters, and they're pretty proud of the trucks and stuff and building their own equipment back in that day. And this picture here, this was gold. I had to go through like 5 law enforcement guys that retired, this and that. I think I got this picture somewhere of us standing out in front of that building. I wanted that building in the background. But that was, I believe it was District 10 Marine Patrol, and there's a lot of these guys that have either just retired or are close to retirement that were associated with that. But an interesting story is that the Marine Patrol there was under the Department of Natural Resources, but there was also a upstart satellite field station down at the West Palm Beach Fishing Club studying different species, sailfish and this and that. And one of the species this group was studying was snook. So they were fishing and tagging snook in the Jupiter Inlet. Somebody reported them fishing and tagging snook, taking them out of the water and stuff in the Jupiter Inlet to the Marine Patrol. The Marine Patrol then proceeded to arrest one of the guys, and then they talked it out, and as they turned out, like, hey, let's all come down to the station and talk about it. During the conversation, the biologists learned that the Marine Patrol were actually going to have their own new building built to spec a little south in Jupiter on Mursiski Road, and that this building was going to become available. And the thought process started going out, man, why waste this opportunity? This is such a cool spot. It's right on the water. We're right here. So it wasn't long, 1988, that it officially became a research facility. Now, at the time, it was under the Department of Environmental Protection. It had left the Department of Natural Resources, became Department of Environmental Protection. But it's been a research facility for the past 32 years, and that's kind of neat and stuff was in there. So now it's here we come in, same Chinese cheeky hut looking building that's been there. If you go there, it's there now, only now it's under the auspices of the Florida Fish and Wildlife. Currently, we have 25 to 27 staff members, and pretty much is made up of 4 different groups. And in there, you can see my group, the group that I'm associated with, is the Marine Fisheries. But let's talk about some of the other folks that are there. First, the manatee section. These guys, especially this time of year, they work. They do a lot of rescue and recovery. The rescues are kind of cool stories, but the recoveries are usually from mortality events. And I'll tell you, manatees smell bad when they're alive, if you can imagine. But these guys go out and they recover these carcasses, and they do what they call necropsies, where they try to figure out the cause of death so that they can calculate if there's an issue, and perhaps there's an issue that needs to be addressed, so they try to put together statistics so we can see how many were related to boating instances, how many were related to cold stress, how many were related to entanglements, and they can kind of determine maybe there's an issue or something that jumps out in an area that we can address to try to stop this problem. In addition to doing those things, they also participate in population estimates, and some of it's, they usually go to areas where they aggregate in the winter times, where warm water stuff, which has traditionally been around power plants and stuff, they've been changing a lot of that. And they also do aerial surveys. On really cold days, they'll go out and fly planes in tight circles all day long. And I thought that would be cool, but I don't know. I was on I was at the fair this weekend, and I was on the kid helicopter ride, and I walked out of that, and I was like, oh my goodness, I can't stand still right now. But anyway, these folks are involved with that. Some of the cool things they get to do is they work with local law enforcement, including our own, but also local ones, to help educate, bring awareness, talk about how they can work together and utilize resources better. But some of the things where you folks might get to see is when they actually do releases, when they catch an animal that's been injured and they rehab this particular animal and then release them. And then most recently, we had in addition to manatees, they also deal with other marine mammals, dolphins, and in this case, this was a juvenile or a baby sperm whale that washed up, I believe it was in Jupiter. And our team went out there. Unfortunately, it passed away, but they were able to get as much information as they could, and also try to pass along. Do a lot of testing that we don't usually get to do. And these guys are especially busy this time of year because this is when there's a lot of boat traffic. There's a lot of people down here. Manatees are on the move, seeking out areas when it's cool, seeking out warm water areas, this and that. So, it's definitely something that is of interest, but it's pretty important stuff as well. Similar to what the manatee folks do, we have a couple groups here that work with sea turtles. One in particular, this is Morgan, who works at our office. She's tasked with rescues, recoveries, nests. She goes out and tracks where nests are so they can mark them so they don't get disturbed and things like that. And back a few years ago when we had that cold snap, there was a story where these guys were going out rescuing hatchlings, and they were riding around with, you ever see those big tubs at Walmart with the yellow lids, black tubs? They had those things full of hatchlings trying to drive away and find places where it was warm, and then once it warmed up, release them. So it was pretty cool. In addition to these folks, we also have turtle folks that work on lighting issues. Some of you guys may be aware that when hatchlings come out, they're driven towards light, usually moonlight, which helps direct them towards the water. So if they have bright lights behind them, street lights and stuff, they can get disoriented and go the wrong way, and that can be a problem. So we have groups that work with people to try to find the right wavelength or spectrum of light so that it doesn't interfere with them, but still be able to have light on their property where they have. And then we also have folks that work with permitting. Zach was talking about how, at his facility they have some turtles that have been permitted. They're lifelong residents. They're not going to be released. And the permitting, kind of the gatekeeper for the turtle permitting, is in our lab, a woman named Megan Kapersky. She's kind of the one who keeps track. Also, if you have educational things like skulls or shells or things like that, she's the person who handles the permitting. So that's another section of folks that work out of our lab. Then we have some folks from Habitat and Species. Tyler Beck, who's at our office here, he is the snail kite coordinator. So he goes around. It's a bird that has had some issues, but he'll go around and they'll try to figure out population size. They'll check out nesting habitat. They'll set up cameras to try to determine which ones are, the survivability and how things are going. But, he spends a lot of time working in and around areas out west where these birds are at. And these guys, I don't know if you've ever seen the big apple snails. Usually, Florida ones aren't that big, but we have some other ones. These guys are specialized. You see their kind of hook beak. They catch these, pull them out, and then they have that beak is used to reach in or poke holes and reach in and pull out meat and eat the snail. And Tyler is also the subject matter expert on the Cape Sable seaside sparrow. Say that 10 times fast. Anyway, that's another bird out in the Everglades that needs some help. Keeps an eye on, and he helps with that, knowing what's going on in different times of year with that species. And then continuing on with Habitat and Species, while these folks don't work directly out of our lab, we have a lot of interaction with them. So the MES group, or Marine Estuarine subsection of Habitat and Species, these guys are really go-getters. They get out there, they work on coastal habitat restoration. So in Palm Beach County or areas like you might have a spoil island that doesn't have any native vegetation, doesn't really have any environmental value, and they'll go and they'll figure out like what level it needs to be at, what kind of plants need to be on it, what the pitch of the shoreline needs to be, what the shoreline needs to be made up of to redevelop it and make it a useful ecosystem environmental area. So they go around doing that. They also, for areas that are good, estuarine or mangrove areas, they'll go and make sure that those areas are being protected, maintained, and manage what goes on in terms of habitat and water flow. They're also heavily involved in marine debris. I mean, I know, we out here, you see stuff floating on shore. Well, in certain areas, marine debris, especially nets and stuff, can be big issues, affect marine life, but also affect a lot of other things. So they work to coordinate efforts to remove marine debris from different areas. And they try to find a lot of solutions in urban areas. One of the key things that they've been focusing on is living shorelines. So in Martin County, when you go off, there's a lot of sea walls and stuff like that. When boats are going, you get a lot of wave energy back and forth, and that wave energy doesn't really dissipate. It hits one wall, and then it travels across, and it hits another wall. Well, if you build in areas where there's living shoreline, where you have trees or mangroves and stuff, and then a sloped shoreline, that wave energy will dissipate as it passes back and forth, and you also are creating a habitat system for other fish and other animals to recruit to. So it's a win-win. It's just finding areas to implement that, so trying to find eco-friendly solutions for problems like that. A lot of their work relies on partnerships, either with counties or organizations and stuff. So they're out actively seeking funding or stuff, or working with different groups. Sometimes it's in-kind service and it's just sweat equity to help get some of these projects off the ground. And they also have a big education and outreach component where they go out and talk to people and try to figure out different solutions to complicated problems. Which brings you to the section that I'm in, which I think is pretty cool, and I hope that when it's all said and done, you guys think it's as cool as I do. So in marine fisheries research, the goal is primarily to do population assessment. So we're looking at primarily commercial and recreationally important species. However, there can be derivatives, bait fish or things that are associated with that, but things that we have control over. And one of the things we look at is life history and ecology. In terms of ecology, we're kind of looking at habitat usage and interactions between other species. But with life history, we can get to a little more detail, and we're trying to look at like age, growth, reproduction. In terms of age, this, some of you guys might be aware of this, but for some folks that I tell that aren't aware of this, they're always usually pretty shocked. The way we tell the age of the fish is, especially with bony fish, we remove their ear bone or their otolith, and it's a structure that's built with a protein matrix, calcium layer, protein matrix. That's a lot of stuff, but basically it makes rings like a tree, and we can count based on how dense the rings are packed together when during slow growth periods, and how during fast growth they're wider apart. And that's usually a seasonal variation, so that equates to years. So for this otolith over here, I know my laser pointer is probably not doing this justice, but we have one dark band, two dark bands, and a margin. So that fish is probably somewhere in the 2.5-year range. Now we can take that and correlate it with growth, with length information, determine how fast the fish grows, at what age it becomes mature, and when it is reproductively active, and when it has the best potential, meaning when can it produce the most, when is it the most important to a particular species. And then we also take into account mortality, whether fish are dying from or being removed from natural causes, natural death, or fishing mortality or incidental mortality. And then we weigh that against how many fish are coming out of the system, mortality, versus how many fish are coming in the system with recruitment. And then we want a higher level of recruitment than we have mortality to create a sustainable system to where you can catch X number of fish, there's enough new fish coming in to replace them, and and that population is going to be stable over the long term. And all that information is kind of put in for overall health and established trends. And we look for, we want to see increasing or stable populations. And for the most part, I think the number is something in the 95% of all species that FWC actively manages are meeting or exceeding their management level. So far, there's been a pretty good job in terms of keeping fisheries where they need to be. I mentioned reproduction, and it's like, what's up with that? So we also do stuff where we look at histology. We'll take little sections of gonad tissue, either ovaries or testes, and we'll see at what stage of development it is based on the cellular, the kind of cells that are present in these tissues. And we can tell whether or not it's male or female. We can tell whether or not it's mature or immature, and we can tell based on the advanced stages of some of the cells whether or not it is spawning capable or actually engaged in spawning. And all that information helps us time the year and the time of day and season stuff in terms of reproduction. So how do we get this information? Well, one of the methods is through a group that's our fishery dependent monitoring group. These guys go out and they rely on the public or groups within the public to provide them with scientific samples. So that could be commercial fisheries, that could be head boats, that could be charter fishing, that could be recreational anglers, and they'll go out and get as much information as they can. They'll meet them at docks, they'll go do interviews at boat ramps or out on the beach talking to people to try to get ideas of how, what they're seeing out there and what's being caught. They also have folks that go to fish houses. So they'll look at the catch rates at fish houses. And then there are folks that are either it is mandatory or voluntarily fill out logs or trip forms so that we can kind of keep an idea on the pulse of what's being caught and weigh that versus what's being seen in other sectors of the fisheries. The next one, this is one that I'm involved in, and this is one that I have a fair amount of interaction with people. This is our fishery independent monitoring, and this is one aspect, but this is what we do out of the Tequesta lab. This is our 600-foot seine, and basically it is a random monitoring program. A lot of folks see us, the engine, the common question I usually get is because we're deploying nets. We have an engine in the middle of the boat, like, hey man, why is that engine in the middle of the boat? And we tell them, well, we're deploying nets out of the back. But what we do is we typically will drop two people off on the shore, holding a net, drive the boat offshore, and make a little loop and drop two more people off. We'll then pull the net in. Everything that's in that net will be counted, identified down to the species, and measured so that we know, approximately what kind of snapshot is in that area. We also take into account the environmental parameters and some of the habitat parameters. Is it a grassy sea bottom? Is it a muddy sea bottom? Is it a mangrove shoreline? Things like that. We take in salinity, water quality, all those things on that snapshot. And then that also can be used to build a picture of what the fishery health is in there. So it's another kind of piece of the puzzle or line of evidence to show what's going on with fisheries. And then comes the subsection that I'm directly involved in, which is the marine fish biology. And this is kind of a hybrid, and I like to say this is, we're kind of the Navy SEALs of this group. We get to do both fishery dependent and independent stuff, plus we do a lot, well, the other ones are focused a lot more on monitoring, and they have a research component. Our primary stuff is research with a little bit of monitoring. But we do a lot of stuff that's involving either inshore, we work offshore, we work underwater, we work with different species, some commercial, some recreational. But that's kind of the gist of what we do. Now, what I want to do is break down some of the projects and some of the things that this group is involved with. All right, so kind of giving you an idea, I'm going to introduce a few things to you so that you can kind of get a taste of some of the projects that we're involved in. First thing I'm going to introduce is acoustic telemetry. Then I'm going to talk about a couple projects that we currently have been going on utilizing acoustic telemetry. I'm going to talk to you about a project that's up and coming, and then I'm going to introduce you to a reef visual census or a fish survey, and then some other aspects that we're involved with in the marine fish biology section. So first, acoustics. So acoustics there's different kinds of acoustics, but the ones that we're talking about are tracking, where we have a sonic transmitter or a coded tag that transmits a signal, and then we have a specialized piece of equipment or receiver that can hear and record that signal. And typically, these little tags here can be detected within 500 to 1,000 meters of a unit. And each signal is unique, so we can track it down to individual species. We can either attach these externally. There's some which is a little less reliable, or we perform little surgeries and we insert the tags into the body cavity, which is internally, which is a lot more reliable in terms of retention and longevity of the tag. And then from that, we can get position information. So here's a project we had involving snook. Here's our acoustic receiver, and here's one of the fish with our tags in it. So all these fish have to do is swim within 500 meters of a tag, and when that transmitter pings, it records that. Then ultimately, we end up with these abacus charts that give us a tag number, detections on a particular station, and we can almost real-time track the movement and habitat uses of fish, and we use this for targeted species. So that, in a nutshell, is kind of how acoustic telemetry works. So moving on from there, we participate in, or we ourselves have built a network of acoustic receivers so that we can kind of create like a grid or a coverage area to where if we're in and around the St. Lucie River, Southern Indian River, we've got a pretty good network of bottlenecks and choke points where we have installed acoustic receivers. And that's both near shore, inshore, and offshore. And we have some representation up in up in Sebastian, Fort Pierce. Our biggest cluster, because we're operating out of Martin County, is in Martin County. But we also have some spread out throughout the southern zones as well. Currently, in our core array, this is our main body of our array. We have 68 receiving stations. We cover about 80 miles of coastline, and the majority of those are offshore, 36 with 24 inshore. Now, this isn't always the case. We're constantly evaluating and seeing where we can get the most bang for our buck, so sometimes we shift things around. But currently, we have a greater need to have things offshore, and I'll get into that in a minute. But as I mentioned in the St. Lucie area, we also have some connectivity with the local reefs and the river, so we have a lot there to kind of pick up fine-scale movements. And then recently, I know it's hard to see in this map, we've installed some gates, which are lines of receivers that run perpendicular to the shore to kind of pick up on highly migratory fish that are just traveling along the coastline versus hanging out in a particular area. And then these yellow dots on our map refer to embedded temperature loggers. So in addition to picking up fish, we're also getting a continuous reading on temperature. So we can pick up cold water upwelling events. We can pick up different factors that are associated with temperature in there, and it kind of helps to correlate. If we see movement and we can correlate it with some kind of temperature drop or some kind of temperature rise, it helps kind of tell the picture better, tell the story better. Now, in addition to our core array, we've been expanding a little bit, and some of this is project specific, and some of it is, related to partners. So, the project specific stuff, I'm going to talk a little bit about Cobia in a minute, but we added a new line of receivers up in the St. Augustine area to help pick up or increase our detectability of cobia that are highly migratory fish. And then opportunistically, we dropped a couple of receivers off of Pensacola, just as we were working with some commercial anglers, or actually some sportfish anglers that were targeting, had events going where they were targeting cobia and they weren't seeing as many. So we opportunistically went out with them and were able to sample a few, but we didn't have any receiving equipment up there, so we put 2 out there, and actually we have been getting quite a few detections from that equipment. And then the last extension here is we've been working with a partner, the West Palm Beach Fishing Club. They've been deploying this artificial reef system called Reef Darts just on kind of central Palm Beach County, which are in deep water deployment areas, either 150 feet or over 500 feet. And we've been partnering with them to drop receivers in there so they can monitor what kind of fish utilization and if any tag specimens are traveling in and out of that area. So those are kind of neat things. And in addition to our array, we partnered with a bigger network of folks that include other universities, other state agencies, other government agencies that have created an even larger network. Zach and I were among the first 2 folks when we started this process back in '97 called the Fact Array. And at the time, it was a handful of biologists sitting around a table thinking, hey, man, my equipment will detect your fish, yours will detect mine. Can we work together and figure out a way that we can detect everybody's thing, and maybe we can spread things out? Well, that's where the idea was hatched, and then more people were getting involved, and eventually it went from, in 2007, where we had a relatively low number of animals tagged, low number of biologists, to, it's grown exponentially over the years, and now we have coverage all the way up, to the Carolinas and beyond, down through the Keys and out into the Caribbean, which creates a huge network and ability to track fish, which is awesome, and it's a sign of like how much just a little seed of something being planted can grow, and a lot of other people can participate and benefit from that. All right. So I mentioned cobia. So one of the acoustic projects that we're currently involved with is a multi-state agency project. Now, in addition to acoustics, they're also using some really neat technology called satellite tracking stuff. And this equipment, like it measures photo period, so even underwater, it can tell what latitude it's at, and things like that, based on where latitude and longitude, based on photo period and things like that. And then eventually it pops off and transmits its information back to a computer or something like that, so they can use that in conjunction with acoustic telemetry. Cobia are both commercial and recreational, and as I mentioned, they're highly migratory. They travel great distances. We've had fish that travel from Louisiana up to the Carolinas and back. So they're definitely movers. And one of the questions that we're looking at answering with all these other partners is determining where the stock boundaries are. There's a Gulf group and there's an Atlantic group. And trying to figure out where that actual boundary is. And ultimate goal to help improve management of the species. Now here's some sciencey stuff for you guys. First off, I mentioned the boundary. So back in 2012, they had the separation between the Gulf stock and the Atlantic stock down at the Miami-Dade Monroe County. And after doing research, they determined that, hey, you know what? That's not actually right. We're seeing a lot more mixing of Gulf fish up and down the coast. So in 2015, they moved that boundary line to the Florida-Georgia line. And then to kind of back that, the rationale behind that, they ran these analyses called network analysis. And basically it just looks at the relationship of one fish to one area versus the relationship to another area. And in doing so, you get these nice plots. So basically the blue and green are fish that were released in either South Florida or Central Florida. The red and orange are fish that were released in Georgia and South Carolina. And you can see while there is some interaction, the two groups kind of do separate. And if you look at it kind of in a different view, there are scatter plots or associated plots, you can see the same story. So we're feeling pretty good about ourselves. Like, hey, that was right. But then in doing so, we also realized that there were some significant data gaps. And when you saw that array that was put into St. Augustine, that's because we identified an area near the Florida-Georgia line, which was known as the zone of uncertainty. We didn't have any detections up there, and there were some questions as to whether or not there was more mixing than we were finding out about. So that was one of the reasons why that was deployed. And hopefully over the next several years, we will get enough information to either verify or give us a good enough reason to shift that boundary line around. But that's an example of how acoustic telemetry is being used to help manage a species. The next project we're involved with is one involving Goliath grouper. Now, I want to give you a little bit of background because some of you folks may or may not know about this, but this species was one that was real common in this area. It's still, it's been making a comeback, but at one point in time, it was perfectly acceptable to catch them and leave them hanging up and everything like that. They were really big fish, so it was a really big catch, but that resulted in the fishery nearly collapsing. And in 1999, they closed the fishery, and then in 1994, they listed it as critically endangered. And what I have here on these graphs right here shows you the red arrows show you what time that the closed fishery took place, and you can see that since that happened, there was some recovery. These are juvenile fish that were caught in and around either the Everglades, the west coast of Florida, the east coast of Florida. This is a plot for adults that were being caught either offshore, west coast, east coast, or around reefs and stuff like that. You can see that they made a big swing. But in 2010, we had a week and a half of freezing temperatures, and that quickly reduced the amount of fish being caught. That doesn't necessarily mean that species, that the overall species took a nosedive, but at least in catching, it showed there was a significant drop in that. So what that shows is that these fish, even though they're big and they're hardy, are sensitive to environmental changes, such as freezes or red tide events like what was going on the West Coast last year and has been going on periodically. So that's one of the reasons that there's a lot of unknowns. So that's one of the reasons why, even though this is a closed fishery, we want to learn more about it so that we can better figure out, hey, how would this fish ever be managed, or is there any reason to ever change the management rules on this fish? A little bit about the background. This fish starts off as a young a year or larval juvenile settling out in mangrove areas. It eventually, as a juvenile, starts traveling out to more structured areas, bridges, docks, or more complex structure, and then eventually makes the trek offshore where it joins the spawning population and participates in spawning, and the cycle continues. Up until recently, the only documented spawning aggregation was in the 10,000 Islands, west coast of Florida down there. And then Chris Koenig actually was able to confirm that we have large groups of Goliath grouper right here in Martin County and Jupiter area, northern Palm Beach County, that are spawning. So that's another reason why we were looking at, hey, let's take a closer look at that fish and see how things are going with it. So we kind of have a multi-project thing going on involving Goliath grouper. The first is a genetic survey, and it's kind of cool. It's a statewide initiative, but our group's primarily working here on the East Coast. And the idea is that we're going to rely on some they call some citizen anglers and things like that to help provide. Instead of us going out and collecting all the samples, we've been working with a lot of folks, crabbers, commercial folks, guides, recreational anglers, and we provide these folks with a kit. And we get them on a special activities license. We provide them with a kit so when they catch these fish, they can just take a little toenail-sized clip of the fin, put it in a vial for preservation, release the fish on its way, and then we have that we can take and analyze the genetic material in that little fin clip. And what's cool about that is there's a couple things that you can do. We can look at the relationship between fish spawning off of our coast versus fish spawning in the 10,000 Islands region and see how much connectivity there is between those two. And then there's another form of analysis called a close kin analysis. And based on the genetic diversity of the between different samples, they can extrapolate that out to and estimate what the population size will be. So that's kind of the gist of this project. And like I said, we work with a lot of folks and getting a lot of assistance with it. But it's kind of neat to see where we're going with this. Our goal so far, we have over 200 samples. Our goal is over the next couple of years to double that, and then with all the other regions that are collecting samples, to turn that all in together and kind of see big picture what we have going on. Second part of this project is the acoustic telemetry phase of this. And one of the things that people have done a lot of work offshore with some of the bigger Goliaths. They get 6-7 feet long sometimes, out there doing their thing. And there's a lot of divers out there that can regularly report on what they've seen, or hey, we notice that they're doing this or their behavior. But as far as inshore, it's a lot more unknown. We know they're in there. We know they need that system as part of their life history strategy, but we don't know a lot about where they are and how long they stay in and all that other stuff. So what we've been doing is we've been targeting fish between 500 meters, so about 1.5 to 3 feet, that size fish, and we've been using 7-year tags, so tags that are going to transmit for 7 years. And hopefully what we will see is fish that migrate from the inshore areas to offshore areas, and we'll see what habitat areas they utilize along the way, and then we can use that information to see, like, hey, this is essential habitat, we need to protect this, or these are areas that we could add more habitat or living shorelines, things like that, to complement the data that we're getting from this. To date, we've tagged 30 fish, and it's kind of been, a lot of it has been in the Martin County area, but we've done a few up in Fort Pierce, and we've done a few down in Jupiter. So kind of giving you an idea, even though this project is relatively new, and new in science, it's been going on for a couple years, but to kind of give you an idea of what we're seeing. When we originally set this out, the thought was these fish are going to stay put, they're not going to move, we're not going to see hardly, much activity until they reach a certain size, and they're going to bolt offshore. And we saw some of that, but that wasn't really the story we saw. What we saw is that there is a fair amount of movement, and there were some environmental triggers that seemed to correspond with some of this movement. In terms of the river, what we saw is when there were sharp drops in salinity. The red line here represents salinity, and the green line is temperature, and the blue line represents the distance, 0 being the distance from being the inlet, and the higher you go, the further upstream you get from the inlet. So what we can see is in times of high salinity, we had a fish, this is the case of one fish, but a pattern held true for several fish of this thing, is that when the salinity dropped, the fish went from somewhere around 13 miles upriver, dropped down to the 6 mile, or 5 mile area, hung out there, kind of oscillated back and forth, and then when the salinity went back up to normal ranges, it went back up to the area that it was originally at, where it hung for a while until the salinity had another sharp drop, where, when that happened, the fish also dropped down and went away, and then when the salinity had a slight uptick, it went back up. The reasons behind that are, there could be a lot of things. It could be associated with tolerance to salinity. It could be available food stuff. There's a lot of factors that are coming in, but it's interesting. And over the course of this study, it'll be cool to see how much interaction. We have seen fish that have been gone for a long period of time return, and we've seen them travel pretty far distances, and we weren't necessarily expecting that. Now, in the Indian River Lagoon, Eastern Stewart Causeway, 25-cent Bridge, that kind of area where we've tagged fish, we didn't necessarily see the same fluctuations in salinity and the same movement correlated with salinity, but what we did see is temperature fluctuations that may have triggered movement. And we've seen this in a couple fish, so we can't say, emphatically this is what's happening, but it's interesting. So we have our red line, which represents the temperature, and we see that it's kind of going around, and when there was a sharp drop in temperature, that's when we had a fish that was at that bridge take off and move offshore. Now, that could be something, that could be an environmental factor that has always been there, and then that's the cue that ends up pushing some of these larger fish or temperature drops. But it's interesting, and we're going to see how that plays out, and hopefully we'll see that over most of our fish. We'll see that they, eventually make moves in that direction and see what environmental factors may have contributed to that movement. I mentioned that we have some new projects coming online, and one of them is pretty interesting. So we have a fair amount of work going on in the St. Lucie River. And we're in Martin County. Most everybody here knows, lake drainage issues, freshwater drainage issues, and a lot of things that affect that. Well, there's been some traction over the years and also some improvements in the water flow system, reservoirs to hold water, mechanisms to transport water out of the way that hopefully they're making some improvements. And what we want to be able to do is track via Fisheries as to how what kind of progress they're making on the comprehensive environment or Everglades restoration program. So in addition to the Goliath grouper and in a previous study that we did down the road where we looked at other species, we can take that data and we're hoping to correlate that with a new project that's going to be targeting spotted sea trout and sheep's head in and around the St. Lucie River and seeing how effects on freshwater flow move these fish. So hopefully we'll be able to build some kind of a resilience and response to these events and be able to show linkages to how it affects fisheries in the area. And we're also going to take into about the other things, seasonality, turbidity, nutrient loads, things like that, and kind of see how all those things play into this overall process. Moving on from acoustics now, I wanted to share a little bit. I mentioned about the fact array, how it's a network of different agencies and different groups that are coming together to do research projects or to help aid research. Well, there's another component that there's another group like that we've been working with over the years, that FWC has been working with, and it's known as the Southeast Florida Coral Reef Initiative. And this group is made up of different state agencies, different organizations, and this and that, private stakeholders, general stakeholders that represent different sectors within the public. And their goal was to get together and try to come up with solutions or potential solutions to address things that affect the coral reef, in and around the Sefker region. Now, I say Sefker region, I'm a little bit old school. Since we adopted this language, but since time there has been a rule that changed this area, or basically along the shoreline, to now called the Southeast Florida Coral Reef Ecological Center, or Ecological Coastal Center, coastal area, or conservation area. That this area is no longer referred to as the Sefker region. And as we go along, I had a map of that, but that map had so many things on it that I thought, man, I'm going to keep this simple. One of the projects that we're involved with this is something called the Reef Visual Census, which is actually a habitat and fisheries component-driven area. It targets natural reef areas, and it's a randomly selected thing. So we pick random areas on a map, and different crews go out. There are samples taken from down in Miami-Dade all the way up through Martin County. FWC typically handles the Martin County or the northern sites. And what we do is we go out on these sites, we drop in divers, and then we do fish counts, or we do benthic surveys in areas, and then all that information is plugged into a master database, and they can look for trends associated with visual fishery surveys in and around this area. And it originally started, I think, in 2011. There was a 5-year baseline period, and now it's an every other year process. This year is going to be an on year, so we're getting ready to gear up, and it typically takes place between May and October. So we'll go out there and we'll start doing a lot of this stuff. But it kind of just shows you another aspect of how we work with folks and teamwork to try to get good fisheries information and plug that into the overall system. All right, another cool thing that sometimes cool, sometimes not cool, is our ability for rapid response. Over the past few years, we've had some tropical activity in this area, and when that happens, our team is in, geographically and also with the skills that we possess. We can go out, and in certain areas we can do pre-hurricane evaluations of area, and then we can participate in post-hurricane surveys to see what happened, what changed, what kind of effects that this had on the area. I have this picture of this red grouper right here, and the reason I do is it's pretty cool. There's a local commercial guy who was a member of the South Atlantic Marine Council, Ben Hardick. And back when we had Hurricane Charlie, he noted that a large slug of red grouper came into our area. Now, typically we get a few here and there, but they're not a dominant species out in our reef. But shortly after that Hurricane Charlie, which kind of came in on the east coast, I think around Fort Myers, or west coast around Fort Myers, there was a big push of red grouper in here. Well, when Irma came through, it hit in a similar area, did kind of a similar thing. So he reached out and said, hey, listen, you guys might want to be on the lookout for red grouper in this area. Now, head boats and places like that typically might catch 2 or 3 of these a month, right? This species. Well, we started reaching out to head boats, and lo and behold, they're limiting out. They're catching 20 a trip, and trips are sometimes several times a day. And their social media outlets or their pictures to show them this everyone on the boat holding this and that. So there seemed to be some truth to that. So we were able to mobilize really quickly to start working with these guys to dart tag so that we could track where these fish. We didn't necessarily know where they came from, but we could track where they were going from then on out. And then we worked with a researcher that did eye lens, meaning the lens of the eyeball, that can potentially use isotope analysis to figure out the origins of some of these fish to see if that is true. Now, we were able to tag a small number of fish, and there's still some results coming in from that, but it seemed like that once they were here, they kind of stayed in the area. But we're keeping our eye on it, and I was talking to another researcher, and when I first told him about this, he was like, man, I don't know, that's a little far-fetched. But then he was in a meeting in, I want to say, like Sarasota area, and they were talking about Goliath grouper. And at the end of the meeting, one of the fishermen came up to him and said, hey, listen, I got a question for you. It seems like after Hurricane Irma hit, all of our red grouper are gone. So that kind of made him a believer in that, well, maybe there's something to this. In terms of emergency sampling, like I said, we're in Martin County. Sometimes I give talks and I show, pictures of blue-green algae and stuff like that, and they don't realize what we're talking about, but I realize most everybody in this room knows what we're talking about when we do that. But there are times, even though my group's not directly associated with water quality and managing water flow, there are times when it's all hands on deck. We need samples from different areas. We need this, we need fish from this area. We need all kinds of stuff. So we will mobilize and help get that happen. And that happened with when we had the blue-green algae output, and then it also had when we had effects of red tide that were wrapping around, ending up on our coast. So we will mobilize and try to pitch in and be part of the big team to get that information so that we can address it as soon as possible. My group isn't necessarily responsible for coral reef damage. We work with the Department of Environmental Protection. But again, we're in an area to where sometimes things happen and there's a limited time to which you can go and assess damage. So if there's an anchor drag or a towboat drags a cable, if you wait for a long period of time, pretty soon algae starts growing over the damage. It's hard to figure out where the damage is. So there's been several occasions where something has happened, there's been a report or something, and we were able to mobilize, partner up with our partner agencies and get out and do assessments so that if there were fines or there was mitigation that needed to be done or things of that, we were able to document it and get it done. And being in where we are, it allows us to be able to quickly do that. And then lastly, we do fish recoveries, and I mentioned some stuff with fish maybe associated with blue-green algae, but also we have from time to time interesting things that wash ashore. If we have a species of shark that may be uncommon or something like that, we can participate, help get there, gather biological information so we can see potential cause of death. Also, some of these things are so rare that they don't get many samples, so we can get there, get the samples that they need, and help get those to the labs and stuff that are doing research on those projects. And then lastly, we get to do a lot of fun stuff too, interacting with folks in our education outreach. Last year, South Florida Fair, one of our team members not only was there to meet and greet, but also did a tour of duty as the super manatee there, and we're able to meet and greet a lot of people. And a lot of folks, their interactions with FWC might be on a law enforcement state or might be something else. So we want to be able to show people all the things that we do and help them see how we interact and what we do to help protect the environment. And then we've had the opportunity to work. This is Ngari, a group that takes kids from inner city kids and stuff that may not have a lot of experience on fishing or out on the flats or this or that. And we'll go out there and we'll do a seine demonstration, and then we'll bring some of the species there that we caught onto the boat, and then we'll show them how we measure them, and we show them some of the things we give. We give them an opportunity to interact and ask questions and be in an environment where it's real conducive to learning stuff and having a good time. And then lastly, our agency puts on a lot of different clinics. One of them is a kids fishing clinic, and I believe this one was actually a disabled veterans fishing event where we'll go and we'll kind of be the hired hand. We'll go out and we'll bait rods and we'll work with them and help them catch fish and do different things and kind of help build them and get them involved in being able to participate and take advantage of our natural resources. And with that, I will be happy to answer any questions. We see scamp, but in my experience, they're not as common a species in this area. Scamp are pretty, when we're doing fish surveys, they have a pretty unique tail structure in that their tail fin kind of sticks out like a little streamer, comes in and then sticks out. So they're pretty easy to identify when we see them. I see them, but I've never seen a lot of them, and this is dating back to my diving activity in this area has been since the late 80s. Well, in addition to what we do, I mentioned the folks that work at fish houses. That that's a lot of commercial catch and a lot of things like that. So if you guys are interested, we can always find areas for trends, and if there's a particular species, we can look at the trends of that species, and you can look at it, and there may indeed be that there was a drop off. If you think about fisheries as a whole and how things have been, it used to be that to find a spot, you had to line up that hotel with that little building in front of it, and you needed to see the lighthouse there. Now we've got GPS that can get us not here, but here, so fishing and fishing techniques have become a lot better, and so, certain species like that. Well, what I think is kind of neat, what I found is areas of reef that are very low profile, sometimes they don't even have a lot of structure to it, but may be hard to delineate between sand and reef. Some of those areas sometimes have the most biodiversity and abundance on them, which is maybe because it's harder for folks to find and get to. No, you're welcome. Yeah, the first question was anything dealing with scamp grouper and the decline of scamp grouper. Absolutely. So the question is are jewfish and Goliath grouper the same species? And the answer is yes, they are. There's different works that try to pick names and things that will be kind of kosher with everybody and this and that. So I think that was no pun intended, but that's the that was the push for that. And and it's kind of fitting. It's a big kind of bruising looking fish. So no, you're welcome, sir. Eye lens material. All right, so the question is, how can the isotope analysis of the eye lens material of red grouper help determine where a fish came from? Well, when they look at fish, and I'm not an expert in this in terms of isotope analysis, I know enough to be dangerous about it. So we're going to look, and they're going to pick out certain chemical factors, maybe magnesium or maybe this and that can be equated to different regions, different estuary systems, or different areas. So let's say that there's a high strontium content in this particular fish that correlates with, and I'm making this up, Fort Myers or something like that, to where they may be able to say, hey, these fish have a chemical signal that is very similar to this area right here. That particular, well, not necessarily natal, but looking at trying to figure out origins of an adult fish, because these fish that they are catching are legal size and adult fish. Isotope analysis is also being used. When I mentioned earlier about otoliths and about features on that, they can do issues where they can go in and take samples and analyze isotopes from that, and that would be more of a natal thing, trying to find out where they settled out as larvae, where they nurseryed at, and then but. The level that needs to be done to that is a pretty high bar, high threshold. One of the things that I think would be very interesting is you can delineate between saltwater and freshwater. There are signature patterns, isotope patterns, that are a little bit easier to kind of separate out versus different estuaries or bays. And with these, with some of the fish that we work with, it might be cool to see, when they're in estuarine areas versus when they're in offshore. So you might see an oscillation between fresh and salt and marine water, and then you might see a stabilization of marine water, and that could also correlate to when a fish left an estuarine, area habitat and transitioned to an offshore area. You're welcome, sir. The question is, if you come across a tag fish, in this case a Goliath grouper, should you try to catch the fish but get close up to read the tags? I would, and especially in the case of Goliath grouper, I wouldn't try to get super. I mean, if the numbers are big, the kind of tags we use, I'll see if I can back up to the kind of tags we use are these streamer tags. Okay. So more often than not, you're not going to be able to read that tag unless you've got 20/12 vision, like my friend in the back room, unless you are physically possessing that fish. So that's kind of more of you catching that fish and be able to release them. I don't know who tags them the way that you see them. And that's a Goliath grouper. I'm not sure who would do that, but I can ask around and find out. But if it was something that was easy to do, Goliath are pretty docile fish, so you approaching them, they're not going to attack you. But if you can read it from 3 or 4 feet away, I'd say look at it. But if you can't, I wouldn't get too close, because they're an animal. You never know what animals are going to do. Well, Goliaths are pretty interesting. They are a very sonic fish, and sometimes when we're diving, even when we don't see them, we can hear them. They can expel water so fast that they make a boom underwater. So we'll drop in on a location and we'll hear a series of booms and there's some Goliath in the area, which is kind of cool. And I remember when I first started diving, seeing Goliath was relatively rare. Or it was, when you saw it was like, oh man, how majestic, how cool. And I still think that, but now it's not as uncommon. And sometimes you see some really big fish out there when they're on the wreck, and they can be seeing them on a picture is kind of cool, but when you're in the water with them, you're kind of like, man, that is a big fish. Let me. Yeah, they're bigger in the water, but in general, they're pretty large fish compared to some of the other species in the area. Ma'am? Yes, ma'am. So the question is, how do we use the data that we gather in terms of management decisions? And there's a couple. All right, so you remember when I was talking about age, growth, maturity, and stuff like that? When we figure out at what, how fast the fish grows, at what age it becomes reproductively active, when it has the most potential as being reproductively active. Let's say this the case of a snook. Snook, we have learned by studying them, is one, they're hermaphroditic. Most of them start off as males and transition into females. We know about what age they do that. We know about what size range they do that. And we know when. We know that the larger fish are typically females. We know that at a certain size, the amount of eggs they produce far outweigh the, removing them from the system. So for this particular species, they came up with a slot limit. So you're allowed to catch one fish that's over 28 inches, but once they exceed 32 inches, you no longer can take them. So that's the situation we're finding out age, growth, and maturity was used. In terms of acoustics, back in 2010 when we had that hard freeze, there were a lot of fish that died. At the time, we were running an acoustic project that was focusing on snook. We were able to provide information to the commission and other managers that over 75% of the fish that we had tagged survived that freeze. We're moving around and doing stuff. That information was used to help open the season on the East Coast earlier than the information on the West Coast. So that's another thing. But that holds true for almost all species. When you have a size limit, the reason that their size limit is this, that we've determined that at this size, it needs to be at least this size to participate in reproduction and be contributed back to contribute to recruitment, replenishment of the population. And then sometimes when it gets over a certain size, it's such a valuable fish that we want to protect that as well. So that's kind of how that plays in. Does that help answer your question? Awesome. Ma'am? Well, the question is, with a giant grouper, would that feed a lot of people? And what we found with Goliath, so I'm going to talk about Goliath grouper because some large groupers don't have the same issue, but in analyzing tissue samples from Goliath grouper, we found that they have very high mercury contents. So in essence, once fish, reach a certain size, it becomes like, yeah, you can eat it, but, it wouldn't be good. So for some other species that may hold true. I know for other grouper species, it's not as big as an issue. But, with that particular species, you wouldn't want to, but for some larger one. But there's a time and place and everything like that. I had a question over here. All right, so the reason I'm repeating is that sometimes some folks have a hard time hearing questions. The question is, what do we know about survivability of fish that have been hooked and released, sometimes with the hook staying in? So there's been a few surveys or a few projects that have gone on. I've been involved with one that was involved with snook a while back, where they would catch snook, tag snook, catch a certain number of fish, and then we would go back in and remove said snook a day later, a week later, a month later, and we would look at the effects of how those, how that would be, and determine whether or not that affected their spawning behavior, whether or not that affected their feeding behavior. And with that particular survey or that project, we found that it had minimal effect. But we do see, like in deep water areas where there's going to be a bathymetric change, or depth-wise in terms of pulling fish up and air expansion, that it's better to use circle hooks and certain things, so you don't, if you hook a fish in the stomach, there's a chance that you're going to cause enough damage to cause mortality. So that's why there's a big push, and in some areas mandatory, to use circle hooks. There's also a big push to use release devices. So if you pull a fish up and it has exceeded its air volume or something like that, there's been, there's kind of been a change in that. They can quickly bring that fish back down to the depth that it needs to be at before it's released. So that's another thing. So there are studies that show, like, some of that handling, if you bring a fish up from 80 feet deep as fast as you can, there's a strong chance that fish may not make it. But if you can quickly get that fish back down to that depth, he may be able to recover. And if you use a hook and you gut hook it, you're going to have a higher mortality rate. Any other questions in the back? All right, so the question is about how do we get the data from acoustic monitoring? And technology is changing, but I'm going to tell you most of it's, in the field, sweat equity. We go out, we dive, we have to go down and service or swap out equipment. There is, as technology changes, there is new equipment that will translate via telephone signal or something like that. But the cost ratio between, potentially a $1,500 unit versus like a $5,000-$6,000 unit, right now it's more cost beneficial to go out and actually do the downloads. But as things change, I mean, I remember I got this old camera that's still on our property list, and it's on our property list because it must have cost over $1,000. And this camera was bought like back in the mid '90s or so, and at the time it was probably the state-of-the-art camera. But now, like, there's cameras that are like $50 that are 10 times better than this camera. So the thought is as technology advances, there will probably the cost of that higher, the higher output or higher technology equipment may come down in a reason where it makes sense. We are doing some stuff. We have basic acoustic equipment which hears something and you download it and it tells you what was there. There's new equipment that has embedded within it tags that send out signals so that you can set up an array that if a fish swims through it, not only will it tell you that fish was there, it'll tell you where it is in relationship to the grid of receivers you have. So that's what they call a VEMCO positioning system, which is some stuff that we're starting to bring online for fine scale movement. But again, that's technology that a few years ago was like, man, this costs too much, and now those prices are starting to come into where we can afford to have a few of those.
Alrighty, guys, let's give Erick a big thank you.
Thank you, guys.